Good morning. Welcome to Whalefest Monterey 2018. My name is Steve Elzey. I'm with the CBS affiliate, KION Television, and it's an honor to be here. I've been here. This is my third year, and it's such a blast. And I like this part of the Whalefest the best because I can learn a lot. Let me talk a little bit about Elan Porter. He's a PhD candidate up the road here at Stanford's Hopkins Marine Lab. And his focus right now on research is improving the understanding of open ocean food webs. And we're going to ask you questions about that, I'm sure. Okay. And I want to say something about questions too. Okay. Through the development, he's researching the food webs through development of cost-effective sampling technologies. Big fish eat little fish. Is that close? Yeah. I could never be a PhD, obviously. But let's
What am I doing?
Give you a little credit at the bottom, would you? But ladies and gentlemen, please, a warm Whalefest welcome for soon-to-be Dr. Porter.
Thank you for that lovely introduction. I'm really happy to be here. It's nice and refreshing to step out of lab and look into a dark room that I assume is full of human faces, and to give a lecture, but also to have a discussion. So today I'm going to be talking or providing a window into open ocean ecology. And one of the things that I like to do at the beginning of these talks is sort of help us as terrestrial animals orient ourselves to life in the ocean. So typically when you look out on the ocean, this is what you're seeing, right? It seems pretty invariable, maybe. There's not much going on, and all of the biology, or most of the biology in the ocean, is happening out of sight. So even when you have whales and mammals that need to breathe at the surface, they're at the surface for not that much time, and even when they're at the surface, the chances of seeing them are really poor, except here in Monterey Bay. Go whale watching, you'll see whales, for sure. So is this really an invariable habitat? And the migrations of predators in the ocean, or of animals in the ocean, suggests that it really is not. And so this is just a map, and it's very cluttered, but every dot on the map is a location of an individual organism, and it's whales. Different colors represent different species of whales, and birds, and seals, and fishes, and turtles. And the main takeaway from this map is that these open ocean animals use a lot of the open ocean. They're moving between habitats, and it's one of the reasons to move from place to place is because you need something that you don't have in the habitat that you're in, right? That there is some variability that these animals are taking advantage of. So that's what we're going to discuss today, and this is the outline of my talk, and it's set up into 3 vignettes separated by time for questions and a little bit of discussion, because I know this is a long slot. We're going to start by just doing a little introduction to biological oceanography, focusing on how physics and chemistry structure oceanic habitats. We'll then discuss open ocean food webs and what that looks like in terms of how predators are looking for forage, for prey, and how those prey are trying to avoid predation. And we'll pay some particular attention to how the unique physical habitat is structuring the unique biology of the organisms living in open ocean habitats. And then we'll talk a little bit about my work, some of my work at the end, looking at a biological sampler of midwater communities, and that's using a fish, basically, to discover the behavior of midwater communities, these communities of forage organisms that are living away from the surface, completely out of sight. So if you only learn 1 or 3, and I can count, so there's 4 things. If you only learn 4 things from this talk, it should be that open ocean habitats are variable. The biology of open ocean of the open ocean is shaped by the physical habitat. We know relatively little about the ecology of the open ocean because it's such a large habitat. And studying whale food can help us understand ecology of open oceans. And this is a little bit of a trick to get you into the room. It's not just whales, it's predators in general, right? Many species of predators are sharing a common food resource, and that's one of the main things we'll be focusing on today. So let's go for a swim. Let's dive into the ocean. This is just a map showing the average depth of the ocean, so you have altitude as color. And you can see that the average depth of the ocean is approximately 2 miles, and oceanic habitats account for over 98% of habitable space on the planet. And it's hard for me to conceptualize that, so I think about if I'm holding an average-sized orange, the volume of the orange is habitable space on land, and my above-average sized body is the rest of the space on the planet, and all of that is contained in the ocean. It's a massive amount of space. And exploration of this huge habitat is limited by its expanse, and then the cost associated with being terrestrial animals that need to go on a ship to sort of track across very small proportions of this large habitat at one time. So open ocean habitats are structured by ocean currents. This is a map of the North Pacific Ocean showing the dominant currents. And basically, you have currents forming these gyres, which are these circular connections of multiple currents that sort of demarcate different physical habitats. They separate regions of the ocean that have different physical characteristics. And this is our big friend, the subtropical gyre, which is where a lot of biology in the North Pacific is happening. And the ocean exhibits a latitudinal temperature gradient, very much like land. So this is a similar map, and it's just showing in color how temperature changes with latitude. And you can see that across the thousands of miles going from near the equator to near the poles, you see an approximately, I don't know, it's like a 30 to 60 degree difference, right? And it takes thousands of miles for you to see a 60-degree temperature variation. However, if we look vertically in the water column, and this line is representing, if we were to just take a slice of the ocean along this red line and then flip it so that now we're looking with respect to depth instead of with respect to latitude, you'll see that there is a strong vertical gradient in temperature over a much shorter space. And this is really important. So instead of thousands of miles, you see a 40 or 50 degree temperature change in the upper half mile of the water, especially near the equator. So the point of this is that oceanic habitats are vertically stratified. There's much more structure in open ocean habitats vertically than there is horizontally, and the biology responds to that. There's also a vertical oxygen gradient, which is driven by contact with the atmosphere, right? The surface of the ocean is in contact with the air, and oxygen freely diffuses into and out of the ocean at the surface. So usually you have high oxygen at the surface because of diffusion. You also have plants that are photosynthesizing, and they're producing oxygen. So that's also driving this pattern of high oxygen near the surface. Oxygen decreases with depth as organisms use that oxygen. So you can see, again, the cool colors represent low oxygen and the warm colors represent high oxygen. And you can just see that basically everywhere there's a decrease in oxygen with depth, and that's because organisms breathe oxygen. So how does this affect animals? Well, temperature and oxygen affect metabolism, right? Temperature especially affects how much energy you need to sustain your normal life. And if you're a predator, it also affects how much energy you need to go and catch fast-swimming prey. And this idea of metabolism constrains the vertical distribution of predators. So where an organism can live is dependent on their metabolism, how much energy they need to perform their basic functions, but then also to feed and reproduce. And that's limited by temperature and oxygen. So we can see an example of this in this figure of diving behavior of tunas, or of this big-eye tuna, where we have time on this horizontal axis, and you can see these bars represent white as daytime and black as nighttime, and then you have depth in miles on the vertical axis. And you can see that these tunas repeatedly dive up and down in the ocean, probably to feed, or likely to feed, but they're returning back to these surface waters because they're these fast-swimming animals that need lots of energy for their fast swimming, and they need to return to warmer temperatures so they can produce that energy that they need. And this also helps them maintain their internal temperature for that basal function. And whales do something similar, though they're not breathing water, they're breathing air, but again, mammals, air-breathing animals, are also tied to the surface in a similar way, where they do these long diving behaviors, but must return to the surface to breathe. So light also constrains the distribution of marine animals. When you have white sunlight hitting the surface of the ocean, it takes basically 600 feet for almost all of that light to be scattered and absorbed by the ocean. So less than 1% of the light of sunlight that hits the surface of the water remains at 600 feet, and this is in the clearest open ocean waters. So because light is limited to the surface, photosynthesis is limited to the surface. And you can see that in the distribution of plant material, basically. So this is another similar figure where you have latitude on this horizontal axis and then depth in miles on the vertical axis, and the color is the concentration, an estimation of the concentration of plant material. And I put our little figure showing the extinction of light here, and this represents that 200-meter depth or 600-foot depth where you don't see any more light in the water, and you can very much tell that all the plant matter is limited to the surface waters of the ocean. So the point of this whole thing is to say that habitats in the ocean are stacked vertically. You have the surface zone in the upper 600 feet, also called the photic zone because that's where there's light, that's where there are lots of photons. And then I just put the average depth on the ocean here for reference at 2.5 miles. So the 1% of the sunlight that makes it through this surface zone takes another half mile to be fully absorbed by the water, and that results in this mid-water zone, also called the twilight zone, because it's not completely dark, but there's very little light. And we're going to be focusing a lot on the biology of this zone because it has resulted in some really amazing adaptations to life in this low light region. Most of the action in the ocean happens in the surface and twilight zones. This meaning that the temperature is most variable, oxygen is most variable, light is most variable in these surface regions, and then as you pass through this 3/4 mile, 1 mile depth, there's very little variation in temperature and light and oxygen. And then because of this, plants and large predators, or many large predators, or most large predators, are very much tied to the surface where there is this primary production, right, this plant matter. So, very briefly, or however much time you need before we go on, do you guys have any questions about this physical structuring of the open ocean? Do you like this self-portrait that I took? Yeah. I can't see. Are we all? No questions? Thank you. You're good. Oh my God. Excellent. So let's see if there's something that I want to say if there aren't any questions. No, let's just move on. Feel free, if you have a question in the middle, to raise your hand. That's pressing. Raise your hand, and if I can see you, I'll call you out, because this is very much a discussion, and I know that this is a lot of information, but I'm hoping that this will be useful context for you for the rest of WhaleFest, and hopefully for every time that you look at the ocean to be thinking about, well, what is happening below the surface, because the answer is a lot. A lot is going on. Excellent. So we just discussed before our very stimulating question and answer session that habitat variability decreases with depth. Most of the action is happening near the surface of the ocean, and many or most large predators are tied to the surface because of their metabolic needs. The entire ocean is home to an enormous biomass of organisms. And again, this idea that animals are tied to the surface because that's where the plants are results in this general trend of high biomass at the surface and low biomass as you move away from the surface. So, this is my favorite part. This is a schematic of a cartoonish representation and simplification of an open ocean food web, where you have primary production, which is plants converting sunlight into chemical energy, and then they're eaten by plankton or primary consumers, and then you have this sort of vague mid-trophic group, which are any animals that eat the primary consumers, and we're going to talk a lot more about mid-trophic organisms. And then it ends in top predators. The food web peaks with these top predators. So, oh yes, plankton, these primary consumers are zooplankton, like copepods. These mid-trophic organisms, if you guys are familiar with lanternfishes or krill that whales love to eat, are considered these mid-trophic organisms. And then our top predators are our friends, the tunas and whales. However, our understanding of open ocean food webs is very heavily skewed towards the primary producers and the top predators. And there's an excellent explanation for that. It's because primary producers, marine plants, phytoplankton, discolor the ocean and are visible from space. So we have satellites up in space, and we can take pictures of the ocean and learn a lot about the density and distribution of marine plants simply by looking at the surface of the ocean. For top predators, we care most about the top predators. They're very cute and charismatic. We also like to eat some of them. Super delicious. They support many economies, and so there's a lot of effort has been put into understanding their biology and how we can best interact with them, whether it's exploitation or simple appreciation. However, this leaves a critical gap in our understanding of how we get from primary producers to top predators, particularly with respect to these mid-trophic organisms. Now, this is a new vocabulary word, hopefully, or not hopefully, for some of you, micronectin. So many of these mid-trophic organisms are called micronectin. And if you see a stranger on the street after this talk, maybe stop them and say, hey, Whalefest is great. Whales eat micronectin. Let's talk about it. And then you can spread the good word of micronectin to your neighbor here in Monterey. So micronectins simply describe small organisms, 1 to 10 inches, micro, that are good swimmers. They can swim against ocean currents, which is nectin. They're made up of these diverse and amazing organisms that have crazy camouflage strategies. And this is the thing that got me into science, into studying science in the first place, is just open ocean camouflage. So the main reason is that it was so interesting is because it was so different from what I had come to learn about and love about camouflage on land or in shallow oceans. So what are some of the differences between predator avoidance on land and in the ocean? Let's have a little chat. Do you guys tell me who has a favorite land animal that has amazing camouflage? Yes. The horned owl. Excellent. Birds like owls. You there? Whales have cool camouflage, yes, and we're going to get to them. That's excellent. So horned owls, and in general, and look, here's a bird of sorts. I don't know if it's an owl, but all of these animals are camouflaged against the ground, basically. They're camouflaged against the substrate. So here you have an amazing lizard, or I think it's actually a gecko. I'm not a biologist or anything. And here you have 2 birds camouflaged against this tree. you have a cute little bunny camouflaged to the snow, and then you have an amazing bug that looks like a leaf. And so all of these animals have great adaptations to hide from predators on land, but it's always against something. They're camouflaging against something physical. Here's another example of camouflage. I don't know if you can tell, but I'm under that dog. So I'm pretty good at camouflage. So in the ocean, you see something similar. You see amazing camouflage strategies near shore where organisms are camouflaging against the substrate. Here's a cool flounder, and here's such a cute little seahorse that's in this, I think it's a gorgonian, some sort of soft coral. Amazing, but again, they're camouflaging against something. And then this is just one of my favorite videos, just to show you one of my favorite animals, if this ever goes. Yes, look. This is just a clump of seaweed, of algae, I promise. There's nothing there but algae. Oh my God, just joking. It's an octopus. Amazing. So no wonder I got into science, right? However, in the ocean, in the open ocean, there is nothing to camouflage against. Many of these organisms never touch the bottom of the ocean, never come to shore. There's nothing except for empty water, or relatively empty. Let's ignore like molecules of water and like sinking particulate matter. So there are several dominant morphological adaptations to life in the open ocean. One of them is transparency, and here's an example of some transparent animals. This glass squid. We have a cool amphipod crustacean. I don't know if any of you recognize the alien from the movie Alien. And that this creature inspired that crazy alien. And then you see fishes. So many organisms are transparent. We also see cryptic coloration. So we know that there's not that much light in the ocean, and particularly red light is the first light to get absorbed. And so you see lots of animals living away from the surface that are colored red because there is no red light to reflect in the ocean. And then you also see this counter shading, which is also common on land, but basically you have a dark back and a light belly, and many sharks have this, and dolphins and whales. Right when you're viewed from above, you have a dark back and you blend into the dark ocean beneath you. And when you're viewed from below with the light shining down, you have a light belly and you blend into the light water that's shining down. Bioluminescence is an amazing thing. These organisms literally emit light. They can detect light in the ocean and then emit light at an equal intensity to eliminate their shadows. And these are this is an example of a strawberry squid, and you can see all these little freckles on it are actually organs that emit light. And then similarly in this hatchetfish, you can see all of these light organs that line their belly. And this is, let's see if so this is just an example of how it works, where if you have counter illumination, you can emit light, and if you're viewed from below, this is what you would see if what you were looking for had photophores and could emit light, and this is without counter illumination. So basically the goal of counter illumination is to eliminate your shadow. And there's lots of cool things that animals do with photophores otherwise, like communicate and attract prey and escape from predators. But the most fascinating thing to me is this elimination of their shadow. It's like a superpower. Conversely, we see lots of animals that have huge, weird eyes that are pointing upward. And the main goal of these eyes is to break the camouflage of all these animals that are trying to eliminate their shadows, where basically you're swimming in the ocean and you cast a shadow, even if there's very little light, even if there's less than 1% of the sunlight that hits the surface of the ocean, you're casting a shadow. So you see all of these crazy animals, like this barrel eye fish and this strawberry squid and this hatchetfish, that all have eyes that are looking upwards, trying to detect the faintest shadow of an animal that they would like to eat. So this is, again, the swath of the diversity of cryptic strategies in the open ocean. So not only are they just some of the most amazing, bizarre animals I've ever heard of, they have an enormous global biomass. So if we just consider fishes, micronectonic fishes, the fish biomass alone of micronectonic fishes is over 34 times that of the global human population. This is just a huge biomass of fish. They're all over the world. They just live out of sight. In particular, these two types of fish, the lanternfish and the bristlemouth, are the two most abundant vertebrates on the planet. Have any of you heard of these fishes? Myctophids, bristlemouths? So let's say 5% of us have heard of them, and they're literally the most abundant vertebrates on the planet. So it seems to be like this out of sight, out of mind sort of scenario where we see this lovely blue ocean and it's so calming and my stress just melts away. But there are millions of tons of these fishes living their everyday lives under the surface. Also, very interestingly, they form dense communities. So we can observe these very expansive communities using scientific sonar. This is very similar to, let's say, what whales and dolphins use to hunt, right? They send sound into the water, it reflects off of an animal, and it bounces back and it forms a picture of where this animal might be. So here we have basically, there's a boat that's moving along the surface of the ocean here, and it's sending sound down into the water, and it's reflecting off things in the ocean. So this is time, but also space, because we're on a boat moving in the ocean, and this is depth in miles. And the color represents the density of stuff in the ocean. So we can see very much that there's variable structure to these communities of micronectin. And it looks like, you can see that there's this vertical migration, and this happens every day. So this mass of stuff moves up to the surface at night, and then migrates away from the surface during the day and sort of diffuses a little bit. And this migration is the largest animal migration on the planet. So whales are moving thousands of miles, individuals move thousands of miles throughout the year, but these communities, when taken collectively, even if they're just moving a quarter of a mile, it's a huge biomass of animals moving up and down in the water every day in every ocean on the planet. So why migrate, right? Like, why is this? It seems like an expensive behavior. What's driving this migration? And it turns out it's a balance between finding food and becoming food. So remember that photosynthesis is limited to the surface, right? So most of the food is up at the surface. We also know that many predators hunt with their eyes, right? So they're also maybe limited to hunting in the surface where their eyes can capture enough light to see their prey. So we know that they migrate up to the surface to feed at night, and then hide at depth during the day in the dark to avoid predation. And there's good evidence that this migration is driven by the amount of light. And one of those pieces of evidence is that during a new moon, these organisms, these communities, migrate closer to the surface than they do during a full moon. So it appears like they're following an amount of light in the water that's triggering their migration. This migration turns out to be really important for resource partitioning amongst open ocean predators. So here, just as an example, you have a big-eye tuna in black, and you can see it has a very big eye. This is to scale. And then you have this yellowfin tuna in white. And we know from studies that have placed tags on both of these species that yellowfin tuna feed much closer to the surface at day and at night. And big-eye tuna feed at the surface at night where there's this concentration of food, but have larger and more sensitive eyes than yellowfin tuna and can dive down and feed on this layer during the day. So it's like they're shopping at the same grocery store, in the same aisle of the same grocery store, but just at different times of day. So that appears to be how they're sharing. We know that fisheries use this information on vertical habitat use, or fisheries have used this information on vertical habitat use to drastically reduce bycatch of species like some turtles and sharks, where basically they know that like big eye tuna are feeding at depth during the day, so they set the hooks. They don't start setting the hooks until they're 150 or 200 meters away from the surface, and this has drastically reduced the amount of unwanted catch or incidental catch. So why is this migration important? And it turns out that this migration is a large part of the open ocean carbon pump, right? It's called the biological carbon pump. And it turns out that if you eat at the surface and then poop at depth, you're physically moving carbon away from the surface, right? So the bacteria that are eating your poop, not your poop, their poop, maybe your poop, that are eating this excrement are using oxygen because they're an organism and they need oxygen to produce energy. And so if that metabolism is happening at depth, then there's less CO2 being respired, being breathed into the ocean at the surface, and that means that the ocean is able to take up more carbon from the atmosphere. This is like some crazy, crazy stuff in terms of just by swimming a half mile up and down in the water every day, there are millions of tons of animals that are physically pumping carbon away from the surface of the ocean and allowing the ocean to act as a better carbon sponge. So we know that these micronectin communities, these communities of small swimming animals, have ecological, economic, and biogeochemical importance. However, we know very little about natural variation in their density or species composition of these communities or distribution, and we know even less about how these communities are responding to natural variation like seasonal variation in their habitat, let alone anthropogenic pressures like climate change. So I think this is an excellent point. Now that all of our minds have been blown with the biological carbon pump, does anyone have any questions? Me. Yes, what is up, young sir?
What's your question? It sounds funny. What's your question? Do you have a question about the fish that look like water?
Yeah.
What
A
is it?
fish.
Maybe we try it without the microphone.
Yeah.
What's your question?
Does this fish bite the whale? It does not. I don't think this fish and the whale share a house or share a grocery store. Yes. So no, this fish doesn't. And it's actually smaller. This fish is maybe smaller than me. So even though it's huge on the screen, it maybe gets to about my size. So it's not terribly big. It's nowhere near as big as a whale. It does. Yes?
How did you track the vertical migration there in the slide
Of the tunas or of the...
of the micronectin?
Yes, let's go back to that slide. So essentially, this is a swath of the water column across time. So you're just driving a boat and sending sound out into the water column consistently and then recording how much of that sound comes back. And so the tracking of this migration is just happening over time as we see this dense signal approaches the surface at around sunset and then is concentrated at the surface for these 8 hours between sunset and sunrise, and then there's a mass exodus, this migration away from the surface of this signal, and then they exist at depth. Does that? Yeah, it depends on the pitch of the sound that you're using. And here, because it's it's high to us, but it's relatively low for this technology, the pitch. We're not tracking individuals in this case. We're tracking aggregations of individuals that collectively produce a signal that's larger than any one of them would produce on their own. So because they're so densely associated, they're reflecting a lot of sound. Yeah. Yeah. Yeah, so the question was, there's a cluster of biomass deep at night. What is that? And while I can't tell you what species it is because we are just using one pitch, I can tell you that migration is expensive, right? It costs energy to migrate to the surface, so there's no indication that every individual needs to migrate every day. So these could be species that do migrate, but perhaps they were full and didn't want to expend the energy to migrate. There are also species of micronectin that don't migrate. So this could also just be reflective of a different group of animals. Yeah. Excellent question. Right here.
So you were saying there's not very much research about some of these micronectonic species.
Yeah.
And I was wondering if I were looking for information about a specific species of like lanternfish.
Yeah.
Where could I find information about that specific species?
Yes, excellent question. The question was, where can I find more information? And the sassy answer is Google. The less sassy answer is Web of Knowledge, which if you're in school, your school should have access to. But you can type in the species that you're looking for, and then it will pull up like scientific articles. Something like Google Scholar or Google I'm on Google all the time. I'm a PhD student and I just Google everything. So I think that's an excellent starting point, and you'll maybe like Encyclopedia of Life. There are just some collections of information that are a little bit more curated that you'll be able to target in on. So maybe like Fishbase or Encyclopedia of Life. Sorry, that's a better, more focused answer. Does that, yeah? Okay, hopefully that was useful information. You seem very together and like you would have run out of resources already. Are there any other questions? Yes. Yes, you there. It's typically good, yeah.
How about the fish that light up like shoes?
Yes. What about them? They're amazing, and there are so many of them. And it's not just one species of fish. Whoa! The fish on his shoes are lighting up, and it's just like bioluminescence. Huh? And do you notice that it happens when you stamp your feet, right? So there are animals that are responding to vibrations in the water. So if there's a big fish that's swimming by, it moves the water, and then an organism can detect that and then emit light. Like there are some shrimp that spew out a cloud of luminescent or bioluminescent, I don't know actually what the material is, let's say mucus, and just agree that that might not be what it is, but that's what it's like. And then they swim in the other direction, right? And leave behind this bright, glowing, attractive thing for the predator.
So why do the whales have to migrate?
Yes, that's also an excellent question. And as far as I know, and I'm by no means, I don't know very much about whales, which is funny at WhaleFest, right? But my understanding is that some places are better for feeding, and some places are better for having families. And so like the population that migrates between Alaska in the north and Hawaii in the south feed in Alaska because there's more food in Alaska. The water is colder, there's more nutrients for plants to grow and to support food. And then they have families in warmer waters where you don't have to like put on a coat to go outside. Is that? Excellent. You had a question, sir.
I was just wondering if the descent of that group on that particular example is different than in the ascent.
Yeah, absolutely. So why is the ascent different from the descent? And this is going to be species specific. And also, yeah, maybe, I guess because it's a light triggered thing, it might be that this is just me making up an answer. But I think that there's, in general, ascent rates are slower than descent rates. And my guess is that it has to do with if you're moving up into more well-lit waters that are also getting darker, it's okay to start a little bit earlier and like arrive at your feeding grounds as soon as they're dark enough to feed. And then for descending, you're swimming away from your feeding grounds as the water is getting lighter, and you need to you want to stay at your feeding grounds as long as possible and then move away from them as quickly as possible because you're quickly going to be able to be seen. There's like an inverse. It's like getting darker at your feeding grounds versus getting lighter. Does that? We'll think about it more together. Maybe, yeah. Any other? Yes, let's try this again. More luminescent fish. Let's talk to me, buddy. I mean, I like it too. We can have an air high five. All right, let's, in the interest of moving to talking about my research, now that we have this context for all of us to go on another journey together, let's move on. Excellent. So, we left off with this idea that we don't know very much about natural variation in these communities, let alone how they're responding to changes in their habitat. So what's, like, why? What's the holdup? We've known about these communities for decades, right? We've known about these layers of animals that scatter sound since at least the 50s. Why don't we know a lot more about them? They seem really important. And it turns out that it's very difficult to sample them. So one of the most common ways to sample these communities is with trawls. You drag big nets through the water, but you only catch things that are too slow to avoid the net or too not stupid is not the right word, but like that can't detect the net and then move out of the way in time. So there's a strong bias towards catching things that can't avoid being caught by nets. We also use acoustics, which we've talked about, the scientific sonar. And again, the main issue here is that we have great information about where things are vertically in the water and how densely packed they are, but we know very, very little about what animal produced this pixel of light. We might be able to tell you whether it's a fish or whether it's a shrimp, but there are hundreds and thousands of species of those animals, so we can't tell you exactly what this signal came from. And then there are many, or I guess not many, but there are a good handful of labs that are using video. They're sending cameras down on robots to visually assess these communities. But they use lights, and these robots are loud, and so it's likely that you're skewing the behavior of animals that you're seeing. So like some animals are afraid of the light and the noise and they swim away, and some animals are attracted to the light and swim towards the vehicle, and that is variable dependent on the depth too, it turns out. So closer to the surface, organisms tend to be more afraid of the light, and closer to and deeper, you have a higher number of organisms that tend to be attracted to the light. I'm just going to give a moment for our new friends to filter in. Welcome. Huh? Please repeat all the questions. I will do that. I will repeat your questions. Not right now, everything that's been asked, but just in general. Okay, I got you. So it turns out that all of this oceanographic work is very expensive. It's expensive to go out on a boat and drag nets through the water, or put robots with cameras into the water, or send sound into the water. And relative to the volume, relative to the area and volume of these habitats, it's very expensive to sample them and to get a high enough coverage spatially and temporally to understand what's happening in those habitats. So what we have is very low spatial and temporal coverage. And this leads to a problem, right? And this is the main problem that my work is trying to address, is that this gap in our understanding of open ocean ecosystem that's in between primary producers, the plants, and the top predators, is limiting our ability to effectively manage marine resources, in the sense that we can't do our best job of managing fisheries without knowing about the food of the species of fish that we are interested in eating. And we also can't do our best job of supporting populations of marine mammals without knowing about how their food is responding to change in their habitat. So what we need, and what I would like to do, is develop new tools or novel applications of existing tools to first better measure variation in these micronectin communities, and then examine how variability in these communities is affecting predators. Let's see. So this brings us to our friend, and you guys are right on time for the best part of the talk, because we're going to be talking about lancet fish. And so that's this fish that's been highlighted in white, and this tray is 1/4 of all the animals that were found in its stomach, in one individual's stomach. So what I'm doing, and I'm not alone, this isn't a field that I'm leading in the sense that it's not a new idea, but we're using this predator to sample the micronectin for us, because there are lots of these predators, and they're much better at catching prey than we are. So this is the long-nosed lancet fish. This is footage from Mbari, which is just located halfway between here and Santa Cruz. And it's one of only 2 videos that I know of, maybe 3, of this animal in its natural habitat. It's this sinuous predator that we know the tissue is really watery. So tuna has a very nice texture to it, right? And it has about a 75% water content. Lancet fish tissue or meat is closer to 90% water. It's very much like, I don't know, biting into like a water balloon. Something like that. So the reason that we initially started to study this lancet fish is that it's the most commonly captured fish in the Hawaii-based deep set long line fishery. So we talked about set depths, right? The fishery is setting hooks below where the turtles are going to reduce bycatch, and they're targeting big-eye tuna. And starting in 2005, the most commonly captured fish in the fishery was actually lancet fish. So for every 2 big-eye tuna you were catching, you captured 3 lancet fish. But we know very little still about their biology, and that inspired this project. So we know that they're abundant midwater predators, especially in the North Pacific, but they're captured in longline fisheries in the Atlantic and in the Indian Ocean and in the South Pacific, and they're all over. They're found all over the globe. They're poorly known. What we do know about them is that they're great at catching midwater micronectin, and we know this from previous studies that have shown that they feed on diverse organisms. For those of you who are new to our room, vocab time, micronectin, small swimming animals, is your main take-home message. Can we agree on that? You will go away from here knowing that micronectin are small swimming animals that tunas eat, that swordfishes eat, that toothed whales and some baleen whales eat, that marine birds eat. And what else? Am I missing anything? Let's go with that. Lots of things eat micronectin. And this fish feeds on a very diverse community of micronectin, over 250 species. And perhaps most importantly is that the prey are very easy to ID. So when we look inside their stomachs, we see such beautiful things compared to studying the diets of other animals. So you'll see here's a lancetfish gut contents, here are the gut contents of an albacore tuna, and here are the gut contents of a Humboldt squid. So the squid chew up their food into very, very fine bits, and we're sort of reliant on ear bones of fishes and the beaks, the mouth parts of octopuses and squid for species identification, and it's very tedious, even for me who does it on fairly regularly. For albacore tuna, remember these are swimmers, predators with a high metabolism. They need a lot of energy to live, and so they're digesting very quickly. So you can see it's well on its way to becoming poop, is the technical scientific term for this state of digestion. And then in the lancetfish, you have these pristine fishes and squids and crustaceans. Here you can even see, or I can see, and you'll just trust me if you can't see, is that there's a stripe of pigment sacs that go along the head and eye of the squid. And so just in the first glimpse of looking at this prey item from the stomach, I can tell you that this is Grimaldi-toothus boneplandii. I know the species. What I don't know, it's like a whiptail squid is the common name, and I know exactly what species it is, and I know relatively a lot about its biology, which means I know a little bit about where in the water column it was eaten. So we're studying lancetfish because it's very easy to tell what they're eating. Oh, this is a lovely video of the dissection process, or not the process, but of everything that was found in the stomach. And I think this is on a loop, which is going to make it very difficult to see, it turns out. So they eat lots of things, and they're all in fairly pristine condition. That's disgusting. I think you mean amazing. I think that we all need to adjust our idea of what disgusting is. You don't have to smell it. You just get to look at it on a computer screen. So what we did was a diet study, and this is a map showing the distribution of our samples, and it's a heat map. It's very scientific and bright and fun. And basically, the important point here is that the coverage of our samples is very close to the coverage of the deep-set fishery. So all of these samples come from federally employed fisheries observers that are on 20% of the ships in the fleet, and their job is to make sure that the fishermen are obeying these laws that protect turtles and birds and marine mammals. And they're also recording every fish that comes onto the boat. And for us, you can request for them to collect samples for you. So every cell of this heat map has a different color, and the warmer the color, the more stomachs come from that region of the North Pacific. And then just as a reminder of the physical habitat, these arrows are representing the main currents, the main surface currents in the regions where these samples were collected. So mostly our samples are coming from the center of this gyre, of this current region, but there's also samples that are interacting with the edges of the gyres, which we've discussed likely represent different physical habitats.
Is that long-line fishery or trawl?
It's a long line fishery, yes. Yeah, excellent question. So what we found are spatial patterns in prey distribution, and that seems like not an exciting finding. I mean, we collected samples over such a large swath of space. It's like 10 million square kilometers. But we were able to see patterns in diet across that space. And so some things were more common in the south. So the shapes, all of these shapes, the width of this shape with respect to latitude, which is here, right, on this vertical axis, is related to the proportion of that prey item in the diet. So this prey item was very common in the southern portion of our sampling area and less common in the north. We also saw some prey that had the opposite relationship of importance with space. Some were more important in the center of the gyre, and some showed no spatial patterns. But these were for individual prey items. If we group them, we see that across space there are distinct, and that's statistically distinct, which just means that they're more different than you would expect by random chance, that there are distinct communities of micronectin being exploited by lancetfish across the North Pacific. So basically, their grocery store is changing across space. The grocery store here near the equator is different than the grocery store near Hawaii, is different near the grocery store at the top of the gyre. And that's what we found, that there are 3 distinct communities being exploited, being eaten by lancetfish in the North Pacific. So the question that we're all dying to know the answer to is, what explains this structure? What explains this variability in diet? Is it related to oceanography? We talked a lot about variability in the ocean at the surface with respect to temperature and primary productivity, and then also with depth. So these are just maps showing on the top panels, this is temperature. Can you recognize, here's Hawaii, here's the North, what is this continent called? North America? You have temperature, so the warmer colors are warmer colors. The warmer temperatures, are warmer colors. And then in these bottom panels, we have chlorophyll, which I didn't change for y'all, but it's just an approximate density of plant matter. So you have a higher density of plant matter where there are warmer colors in these bottom 2 panels. So these white boxes are our sample areas, which you can see cover a large range of temperature and oxygen. And we have a winter sampling period and a summer sampling period. And you can see that the physical oceanography changes across seasons. Are we all on the same page? Yes. Ocean changes with season. We got to sample that change. What we found is that the patterns in diet also change with season. So in winter, we have 2 different diets, and these shapes just represent the areas within which lancetfish have similar diets. And if they're in different shapes, they have very different diets. And the main takeaway is that in both summer and winter, there's a border, there's a boundary in diet right at the boundary of the gyre. So there's a current that's separating the habitat on either side of this line at approximately 18 or 15 degrees latitude. And lancetfish caught south of that are eating drastically different things than lancetfish captured north of that boundary. And in the summer, we also see a boundary sort of just above Hawaii. And if we look, so now, right, we're looking at the surface of the ocean. If we, again, take a vertical slice of the water column and look across the North Pacific, so now here, this is just our reference map, and all the blue dots here are places where we've taken the temperature with depth. So we send a machine down into the water and take temperature once a second. And now we're looking from, let's say, 10 degrees north to 40 degrees north. And you can see that this line represents this boundary, right? It's the same latitude that this dietary boundary happens. And you can see that there's a change in the oceanography at depth, where here the waters of 10 degrees are sort of not really changing with depth. And this whole water mass appears to start moving towards the surface right at about 23 degrees north, which is where we see the difference in diet. So here in this upper panel, we have temperature with depth, and here we have salinity with depth. And the point here is just to say that there is something about the habitat, about the physical habitat, that is changing at the latitude across which we see variation in diet. So this is like, I guess this is like Erdley Avenue, where in the Pacific Grove side, you have Safeway, and in the Monterey side, you had Nob Hill, and it will soon be Safeway. I needed a better analogy, maybe. Yes, different diets across a physical boundary. And this seems so simple. There are different habitats. Why wouldn't we expect there to be different communities of animals that are adapted differently, but best to those different habitats? And the main takeaway is that we just haven't been able to get such a large sample coverage to even measure that and to test that hypothesis until now. So what we learned is that lancetfish are good biological samplers. We can use lancetfish to sample micronectin communities for us because they are abundant and readily available thanks to an existing federal program, right? These federal fisheries observers that generously provide us with samples. And they eat poorly known micronectin, which is kind of like a throwaway, or just, it's like a baited statement because most micronectin are really poorly known relative to land animals. And they're easy to ID. Lancetfish don't digest their prey very quickly, and we can see exactly what they're eating. We can detect variation in the diet that we think is representative of the communities of micronectin that lancetfish are eating, right? And these differences in diet coincide with physical structures in the ocean across which we would expect there to be different communities of organisms because the habitats are different. And our conclusion was that this sampling method is a useful supplement, is a useful addition to traditional sampling methods, and will drastically improve our understanding of how food resources vary across open ocean habitats. And that's very grandiose, right? Like, this is so important, it will dramatically improve how we interact with the ocean. But here's why. This type of information can help us know or understand why animals are migrating. Right? We said some places are better for having families, some places in the ocean are better for finding food, but we're making those statements based on sea surface temperature mostly and ocean color, right? The distribution of plants measured mostly at the surface. So if we know about how food for all of these predators, all of these birds and whales and seals and turtles and fishes, if we know about how their food changes across space and time, then we might be able to better understand why they're migrating or what is driving those migrations. It also might help us better understand how food resources are shared between open ocean predators. So this is a figure, and what's important is that each color blob is a different species of tuna. Okay, we're not covering a huge diversity of fishes. These are all tunas. And this is just a schematic showing that with latitude, so here are the tropics, and here are the temperate waters, like around Alaska or north of Alaska, you can see that each species has a set distribution latitudinally, and that they use different vertical habitats depending on where they are in the ocean. And that's, again, related to their metabolic needs, and then also perhaps what food is there. So what happens if the food that yellowfin tuna eat moves north? How does that affect how yellowfin tuna and big-eyed tuna are interacting with each other, right? So the main question we would like to address is that if we can quantify, first of all, what all these animals eat, which is relatively easy for tunas and things that are brought up by the fishery because we have access to them. We can know what they eat and what their preferences are. If we know where those preferred food items are in the ocean and can predict how they would respond to change, we could then predict how changing food distribution might drive changes in the distribution of predators and then changes in how those predators interact. So very quickly, we've just escalated from like, what's here? How does this food vary to how are the interactions between these predators who have very specific strategies for sharing this food going to change as the ocean warms is like the biggest question. And then finally, hopefully this would inform management decisions where if you're building assessments of stocks, right? If you're saying, okay, based on how much hake we caught last year, or maybe hake is a bad example, but let's say how much tuna we caught last year, let's make a decision about how much tuna you're allowed to catch this year. And if you don't incorporate information about how tuna food is doing, then there's bound to be a greater error or a greater risk in terms of making decisions about how much you can remove from the ocean when you're ignorant about how much food is available for the resource you're trying to extract. So, things for all of us to remember. The ocean is the largest habitat on Earth. It is huge. Go home, pick up an orange. The volume of the orange is the volume of habitable space on land. Your body is approximately the volume of habitable space in the ocean. It's mostly in the ocean. It's so baffling because the mountains are huge and we live on land, but 98% of space on Earth that animals can occupy, let's ignore like animals like birds that sometimes stay in the sky for years on end, is in the ocean. The biology of the ocean is structured by light, right? Photosynthesis is limited to the surface. Millions of tons of animals migrate away and to the surface based on the amount of light that's in their habitat to feed on the food that's concentrated in the surface. We saw physical adaptations, morphological adaptations like camouflage strategies that are based on the amount of light in the habitat. And we also know that temperature and oxygen are important for sort of limiting vertically and then latitudinally where animals can live, like what temperature they prefer so that they can maintain their basal metabolic rate. Micronectin are a diverse group of organisms that undergo the world's largest animal migration. They help the ocean absorb carbon. Remember, they eat near the surface, and then they poop at depth, and that physically pumps carbon away from the ocean surface and allows the ocean to absorb more carbon from the atmosphere. And they support commercially and culturally or socially important predators like tunas, whales, birds, other mammals. And then finally, we can use lancet fish as a relatively low-cost tool because they're provided by a federal program that's in place to protect mammals and turtles and birds, right? These are federal mandates that aren't going anywhere. So this program is providing samples basically free to us. And this sampling of lancet fish will improve our understanding of poorly known micronectin communities simply by looking at their diet across space and time and then using that as a proxy for how some micronectin are responding to variation in their habitat. And then just briefly, I'd like to acknowledge the observers who are providing the lancetfish samples to us, my colleagues at NOAA and at Stanford and at University of Hawaii, and then the amazing taxonomists who I've been working with and who have trained me to identify these 250 species of animals that lancetfish are eating. And with that, I'll take any more questions. Yes.
So I was wondering whether more commonly known forage fish are the anchovies and sardines are included in micronectin, or are they different groups?
That's an excellent question. The question was whether more common forage fishes like anchovies and sardines are considered micronectin. And it's an interesting question. Let's say it depends on who you're asking and what the question is. So yes, they fit the definition of micronectin. Let's just say that. Yes, they fit the definition, but they also have commercial and ecological importance that's like pretty high for an individual species and may be treated differently in the sense that we have a lot of information about anchovies and sardines. So if we were to try to predict what's happening to the animals that feed on them, we might have enough information on anchovies and sardines to say, okay, we know what the abundance of these are and how that changes over time, and then we might group all the rest of the micronectin together, if that makes sense. We just care more about them so we know more about them, so we don't have to treat them as a group. We can treat them as individual species. Yeah, excellent. Yeah, John.
So, with all that fishing on tuna, vertically, they do look to stop quite substantially.
Mm-hmm.
And also lanternfish, right? I mean, if you're fishing on them, that's
Lanternfish? Lancet fish. Are you talking about the Yes. Yes. Yes.
So is there any information about what that does to the micro?
Excellent question. The question was, we are removing a lot of predators from the ocean through fisheries. Whether we are targeting the species like tunas or swordfishes, or whether the species are captured incidentally or accidentally just because there are hooks in the water or nets. And one example is lancetfish. We don't eat them because their tissue is really watery and it doesn't taste good. What does removal of these predators from the ocean do to the micronectin? And it's an excellent question. And I don't know. I have basically, there's this idea, right, of top-down control, of that these predators might be controlling in some way the populations of micronectin just simply by removing individuals from the population through predation. And if you reduce the amount of individuals that are getting eaten by predators, by removing predators from the habitat, then you might see this explosion of numbers or an increase in numbers of these prey. I don't know how strongly predation controlled these communities are, especially in the center of that gyre where we saw that here, where chlorophyll is really low. So this is considered to be a desert, and so we might not expect there to be a high abundance of animals that could be so influenced by the predators, but it's definitely something worth modeling, so we could try build a mathematical model to describe that. But it's unclear, right? I think we don't know enough about the normal abundance of these communities to be able to accurately track changes with respect to how many predators are being removed. Does that? Yeah, and a lot of these predators are eating, so remember, like, we don't eat the same thing throughout our lives. Like, I didn't like tomatoes until I was 10, and now I eat so many tomatoes, I'm basically a tomato. And so, you know, like tunas might also, or do also have that change in food preference with size. So we're removing mostly larger individuals of these predators, and so those might already not be mostly feeding on micronectin. They might be feeding on animals that feed on micronectin. So I think it's a complex question and one that we can't really answer without more sampling, or unless you're better at math than I am, and then we can hypothetically try to figure it out. Yes? That's an excellent question. Sharks and tunas. The sexiest answer, and I think that it's an appropriate adjective when talking about fish in general, is that lancetfish eat other lancetfish. And that might be the most interesting, one of the most interesting questions, and it's something that I hope to study when I'm done with my degree at Stanford, is that lancetfish eat lancetfish. And when you're removing lancetfish, or large lancetfish from the population, you're also reducing predation pressure on lancetfish. And so there's this potential for positive feedback, for a positive influence on removal of adults by the fishery on the population, which is basically unheard of when you're talking about fishing, right? It's like, this is very bad how much we are taking out of the ocean. But in the case of lancetfish, it might be a little bit more complicated than that, where you're like removing these crazy cannibalistic adults from the population, and then more juveniles live to adulthood. So there's, yes, it's a complicated question, or the question that I posed to your question. But sharks, tunas eat lancetfish. We do not. They're gross. Yes? Eat, sorry, one more time? Well, the most basic answer is that they digest them and then poop them out. Oh, the question is what happens when the big fish eat so many small fish? And my answer again was they poop. because they're animals. Yes, they need, just like we need breakfast, and maybe you don't eat lunch, like you're weird like that, and then dinner, we need energy in the food to just function as animals, but then also to go on a run or walk around, or if we're studying, we're going to use more energy and we need food. And that's the same reason that big fishes are eating little fishes, is that they need to basically survive, but then they also need to swim fast to hunt for prey and then have babies of their own. Yeah. Yes, over there. This is a lancet fish. I should have you'll just take my word for it. This is a lancet fish. There's a verbal label to it. Yes. One more time? The question was, where does the poop come from? And the answer is their butthole. Fishes also have butts. And it's funny, but it's also true. I love the snout. I wish I could do that, like control the audience with a snout. You there, you had a question? Oops. One more time? Which state? So, and this is a very weighted question. States are societal constructs that they live mostly in the open ocean. So they live all over the world. They're not living in any one state, and they might come close enough to shore to be inside what's called the exclusive economic zone of the United States, where we have exclusive access to resource extraction, such as fisheries, for 200 miles off of the coast. And there are definitely lancet fish within 200 miles of the coast. Sometimes they wash up onto beaches in Seaside and Monterey. But no, they are open ocean animals. They don't care what state they're from. They're not from a state. They're global citizens. We had a two-part question over here. Very interesting. Can we sample the micronectin with DNA sequencing? And the answer is yes. The answer is modified by should we, and what does it actually tell us? So the way that I understand how environmental DNA technology is working right now, and that's the term that's used for taking like a swab, let's say, of the ocean, and then using molecular techniques to say, okay, what DNA do I find here? What species are here? And as of yet, there's no reliable way, as far as I know, to tell you how much of an individual species are in any area. So you can get a list of like, this fish was here, this wasn't, this other fish was here, and it was here in May, but not in June. But it's just a presence-absence thing right now. We're not really able to say, this community was 60%, lanternfish and 20% bristlemouth, and there was one like chubby squid. So we don't, so it's useful for some people for some questions, but not for this yet. We all have high hopes. And I think the technology is becoming cheaper and cheaper. What was your second part? Yeah. Well, I mean, like algae, I probably, yeah, I think it's mostly seaweeds. Yeah, that's an interesting question. Algae, and there are great books on algae like edible algae of California, and like when and where to go like collect it if you would like to incorporate that into your diet, vegan or not. Yeah, right here, down in front. Yeah, I can.
That shows the area where the fishing industry is being served by observers.
Uh-huh.
So they're following rules by fishing legally.
Yeah.
Turtle
Wowza. The question was, in America, we follow the rules. What about elsewhere? Yes, it is a very interesting question, and I am not the right person to give you a knowledgeable answer. My understanding is that a lot of the rules, or not a good number of the rules that we are following, are rules that are specific to America. And we have agreements with other nations in terms of like protected species. But like the number of birds that you're allowed to catch accidentally before the fishery shuts down for the year is very much set by the American government for American fisheries. My guess is that, you know, and this is a tough question, right? Because, there's so much here. How do I want to go about this? It's a tough question because in other countries, but also in America, first and foremost, fishing is providing livelihoods for people and supporting economies. And so, especially when the government is involved, their first concern in all things science with respect to, like, NOAA, the National Oceanographic and Atmospheric Administration, and their study of the ocean is how do we support the people who make their living by fishing, right? And sometimes that means stopping fishing after a certain amount so that next year there's enough fish. But their mandate is to support the economies and the people that rely on fishing. And we're starting to get a better understanding of to do that well, we need to incorporate information from other parts of the ecosystem. But it's very much focused on the people and supporting the people. And I think in other countries, as far as I know, it's hard to like comment on their practices from a standpoint of like, well, we're following the rules because first and foremost, there are people who need to feed their families and make, whether that's fish themselves or by selling fish. But there's excellent work being done on the intersection between fisheries management, social science, and ecology. So how do you make sure that you protect the ecosystem, maximize how much you can take from the ocean sustainably, while making sure that all the stakeholders, everyone who is benefiting from the resource extraction, are getting their needs met, right? Like, conservation isn't going to happen unless everyone who's affected by conservation decisions come together and decide on a method that meets everyone's needs. And I think it's very complicated, but I think that's a field of study now fairly well developed and continuing to develop, and there's a lot of interesting work coming out of that field. Yeah, great question. Way over there. Go for it.
If a large fish is eating a little fish and the little fish dies, or what happens?
If the little fish dies, you mean that individual little fish, or if you mean all the little fish die? Yes, that's an excellent question. So what happens is that either they would choose something else to eat, and that would change, like, either the grocery store that they ate in or the aisle of the grocery store that they shopped in. I mean, that might change how many other fishes that they need to compete with for that one new type of fish, or they would starve and die, right? Like, if you don't eat, you don't make energy, you don't live. So there's two main possibilities there. Yeah, great question. Again. Yes, very interesting. The question, which was very interesting, was, is it true that some sharks eat each other before they're born? And I'm trying to remember, the name isn't important. The answer is yes, very creepy, very cool. Interesting strategy for making sure that when you leave, when you're born, you have a higher chance of getting everything that you need to survive. Is just like making sure there are less babies to compete with. You there? That's interesting. So similar to birds, there aren't two separate places for pee and poop to come out of. I see a theme from this side of the room. It's the same hole. Yes. You there I'm in the back corner. Yeah. What do sharks eat? It depends on the type of shark and the age of the shark. But let's say fishes and squids and turtles and mammals and like sometimes trash, yeah, or like man-made objects. Yes.
Do fish ever get thirsty?
Oh, that is a very interesting question. No, I don't think they have that mechanism for feeling thirsty. I think that they drink water. That's very interesting. They definitely need to maintain a certain amount of water in their body, but they're living in water. So they don't necessarily have to get that water by drinking it, right? By like bringing the water into their stomach, right? They have these gills, which are structures that they breathe across, but water can also pass through those gill structures. So it's maybe drinking in another way. Very cool question. I'm going to Google that and come up with a better answer for next time. That was awesome. Yeah, over here. Yeah, you. The question was, do little fish ever eat big fish? Is that right? Yes. The answer is yes. Oh my God. There's a fish called a swallower. And let's say the fish is the size of this clicker, and it's a deep sea fish. And one thing that's important to know about the deep sea is that there isn't a lot of food around. So when you encounter a burger or a piece of food, even if you're not hungry, and even if the burger might be much bigger than you, you're going to do your darndest to eat it in some cases. And that's the case for the swallower, which sometimes eats food, 10 times bigger than it. It has like a big balloon of a stomach that's very stretchy, and it can fit big things inside of it. Very cool question. Maybe one last question. Yes. When do or what do? Wow, what a loaded question. Why do sharks eat humans? I think first and foremost, it might be good to say that if you were to look at the diet of every shark in the ocean, humans would account for a very, 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 very small proportion of what you find in their stomachs. I think in general, sharks attack humans when they confuse the humans for something that they normally like to eat. Yes, you're like more likely to get struck by lightning than you are to get attacked by a shark. You might even be more likely to get struck by lightning twice than you are to get. That was a fast question. Did you have a question, sir?
Oh, the swallowfish,
Yeah.
does it kill fish larger than it, or
Just swallow them. Yes, I don't know. That's an excellent question. But their stomachs are very flimsy looking. So I imagine if you have a large fish inside your belly that's kind of strong, it might just thrash and alien right out of that stomach. Yes, cool. Well, thank you very much for your time. Yeah. Oh, one last thing. My lab, where I work, is doing, they have a booth where Squids for Kids is our outreach program, and we're dissecting very large squid, like larger than all of you. It's not a giant squid, but it is very large. Yes, go check it out. It's that away. Yeah, thanks.